All right, we'll go ahead and get started. My name's Jim Hotaling. I'm a urologist who does men's health and male infertility at the University of Utah. And we're going to talk about sort of where the future might be going for male infertility. We're going to cover its relationship to somatic health, talk a little bit about the Sperm Olympics, which is essentially a shorthand for microfluidics, and then talk a little bit about in vitro spermatogenesis and how it could impact the field going forward. So thank you for inviting me to give this talk, and hopefully we can keep everyone engaged and also learn a little bit about the field. So in terms of my disclosures, here they are: Boston Scientific and Endopharmaceuticals are educational grants. And then I do own and have founded 3 startup companies: Nanonc, Andro360, and StreamDX. Some of this work, the microfluidic stuff, is directly related to Nanonc, which is the company that has developed this technology through funding from NIH grants. In terms of the learning objectives, we want to talk about how male infertility impacts somatic health, talk about whether microfluidics has a role in male infertility, and then talk about how spermatogonial stem cell culture may actually have an impact on the field. So the first thing we'll talk about is something called SHARE, or Subfertility Health Assisted Reproduction in the Environment. This is a big cohort from the University of Utah that we've used to look at male infertility and its relationship to somatic health. So Utah is a very unique state in a number of ways. It's a beautiful state and has a lot of diverse geography, but it has a population of 3 million, which is the 31st biggest by population, but the 13th largest by area. So it's a very, a lot of it's very rural and not very densely populated, with the exception of maybe Salt Lake and St. George. It's also the only state where the majority of the state belongs to one religion, Latter-day Saints or Mormon. And that's relevant for this database because for 2 reasons. One, the Mormons are to some degree a founder population. They came to Utah in approximately 1860. And obviously there's been a lot of in-migration and out-migration, but for religious reasons, they keep extensive pedigree data. So a lot of the people in Utah can trace their genealogy back to the 1860s, or sometimes even further. And a lot of really important genes, such as the BRCA gene or the APC gene, were actually discovered from this cohort. So this is a picture of the Hubble telescope, and the database we use to do this is something called the Utah Population Database. And a lot of people have described this as akin to the Hubble telescope for population health. And we basically train this thing to look at sperm by linking it to some large databases we have in Utah. The Utah Population Database contains about 20 million records. Essentially everybody's who lives in Utah is in the database. If you have a Utah driver's license, you're in the database, whether you want to be or not. Having this database in other states might be quite controversial, but it's been in place in Utah since the 80s. It contains about 85% of all medical records, and then all birth certificate, death certificate data. It has genealogy data on about 50% of the state, and then a lot of fetal deaths, census data, and driver's license data, which gives you a height, weight, and address for everyone. So a lot of people have shown, Mike Eisenberg's done a lot of this work, that infertile men are more likely to have a number of health issues. And we ask the question of if there is either a genetic or environmental underpinning of that connection, does that extend to families? Particularly when it comes to genetics. So we basically said, okay, you have this infertile person, what happens to their offspring going forward, and other things historically in their family that have relationships with any of this? So like I said, we've looked at the proband, but what if male infertility and the sperm count was a biomarker of familial health? If that was the case, we could really derive a lot of interesting insights from that. It may have broad population relevance, and also there may be some biologic underpinnings of that may give us insight into male infertility, and even cellular division and senescence on kind of a basic science level. So when we look at individual versus familial health, we know that infertile individuals have higher risks of diabetes, heart disease, alcohol and drug abuse. They tend to die younger. They have higher rates of testis and prostate cancer and higher rates of psychiatric disease. You know, I've done some of this work. Mike Eisenberg has also done a decent amount of this work. We also know that from a familial standpoint, these individuals, that their families, this is from our work in the shared database with the Utah population database, have higher rates of testis cancer, thyroid cancer. Their relatives have higher rates of pediatric cancer, acute lymphoblastic leukemia particularly, and higher rates of pediatric mortality and deaths from major congenital anomalies, which really begs the question of, you know, why is this? What is really driving this? So when we look at, you know, what are the conclusions and how does this relate to specific sperm parameters, we see that first-degree relatives of azoospermic men have higher rates of thyroid cancer and death from major congenital anomalies. Men with oligozoospermia or lower sperm counts have higher rates of pediatric acute lymphoblastic leukemia. And all infertile men, their family members, have higher rates of testis cancer. So what's the mechanism? It could be genetic, it could be environmental. It also could be epigenetic, and we'll talk a little bit about that in a second here. So what is sort of epigenetics? Really, it can be summed up by this picture. This is a picture of Darwin and Lamarck. Most people have heard of Darwin, not everyone has heard of Lamarck. And basically, this can be summed up by an analogy to a giraffe. Darwin believed that giraffes have tall necks because their ancestors, the ones with taller necks, had a selective advantage and had more kids because they lived longer because they could reach the leaves. Lamarck believed that giraffes have taller necks because they stretch to reach the leaves, and that changed the genes that were then passed on to their offspring. Lamarck was largely discredited in his lifetime, but it turns out actually he was partially right. So this slide sort of illustrates this concept of paternal preconception health and transgenerational epigenetic inheritance. Here you have these mice that were taught to fear an odor, either benzene or acetophenone. And what you found is when you expose them to benzene, you shock them, and then you bred them out for several generations with really no exposure to their parents. And what you see is that the F1 and F2 generations have an increased startle response when you expose them to benzene, which essentially means that there was something passed through the germline that actually their parents' life experience changed their genes. So here you see not only that, but the gene for an olfactory receptor activated by benzene was demethylated in the germline. Hence you see, you know, behavioral and really truly biologic evidence of this going on. You know, how is this mediated? What is the sperm epigenome? Essentially, the best way to think about this is if genes are like, you know, the words in a book and pages in a book, the epigenome is like how you select for what you're actually reading within that book. And there, the sperm is wound up really, or the DNA is wound up really tightly in the sperm. And you have these, you know, histone methylations, which can change what genes are read and not read based on what's exposed. So when you think about this, you have to look at DNA methylation. You have to look at nuclear protein composition. You know, RNA is another big one. And when you think about RNA, there's this whole concept of microRNAs. And the idea is that as the sperm passed through the epididymis, that microRNAs could actually be coming in and out of the sperm and actually be what mediates some of this transmission of how life course experience gets passed on to offspring, that this is really happening through microRNA. So interesting work here and more to come. One example of this, and we wrote a review of this several years ago, is that obesity is actually heritable. And that's beyond just, you know, if your dad ate a lot of potato chips, that you're more likely to eat potato chips. That's actually that someone who's obese, it actually changes the genes for their offspring. And this is a number of different examples from this from both animal studies and human studies. And basically mice that are fed a high-fat diet, it changes the genes, the glucoregulatory genes in their offspring. So this is all happening through the epigenome, and this affects not only offspring, but also the grand offspring. So some really interesting work here on epigenetics. Shifting gears a little bit, we also use the UPDB to look at, leverage the pedigree data. And these are an example of a fat pedigree and a skinny pedigree. And the fat pedigree, which is not totally atypical of our pedigrees, these people have a lot of kids, especially historically. And the skinny pedigree is a smaller pedigree. And when you look at the population in Utah, especially if you go back to the 1860s, they generally speaking were LDS, and most of them desired very large families. So what we did is we asked the question, is there a relationship between subfertility in one generation and historic intergenerational family size? So in other words, if you have a low sperm count, is your family historically likely to have less kids? And we identified this cohort of families with small intergenerational size, and we looked at data before 1935, which is when birth control was introduced. And what we found was that men with a low total motile count have smaller intergenerational family size, which is super interesting and actually somewhat surprising. So this was our cohort. We had 26,000 men with a semen analysis. We had 2,800 when we got to families where we had complete pedigree, and then they had to have generations before 1935, which whittled this down somewhat further. And then we also had to have made the requirement that they had a measure of total motile count in a generation before 1935. We ended up with about 2,200 men to look at with complete pedigree data. And what we found when we looked at total motile count as a continuous variable is that for every additional child in their historical pedigree back in 9 generations, we saw an increase in the total motile count of 1.88 million that was significant. And obviously you could say some of this could be confounded due to selection, you know, some families wealthier, healthier, other things, but in reality, it's unlikely to have, you know, confounding going back 100 years that persists throughout a family. So we see that lower total motile count equals smaller intergenerational size. Another way to put this is men in the top quartile of intergenerational family size had a total motile count that was 48 million higher than men in the bottom quartile. So really a dramatic difference there. So what's the mechanism of this? So Aaron Quinlan, who's a computational biologist at the University of Utah, looked at big 3-generation pedigrees where they had whole genome sequencing and found that, and this is in another cohort from the Utah population database, but not related, not directly tied to the work that I'm doing. So these large families show postzygotic mosaicism and variability in germline mutation. So essentially what they did is they took 33 large multi-generational families with an average of 8 kids per generation. So big families. They found that the de novo mutation rate, about 75% of which is driven by the male, varies from 0.2 to 3.24 across different families. And what they found was that families with a higher de novo mutation rate, which there was this subset of families that had a 3-fold higher de novo mutation rate, had a lot higher rates of cancer, and they died on average 8 years earlier. So, you know, it sort of begs the question, is it possible that these skinny pedigrees have a higher de novo mutation rate? And we certainly don't have proof of that at this point, but it is a plausible reason for what we're seeing in the epidemiologic and demographic data. And we are investigating this and have applied for some grants to get funded. The whole genome sequencing, as you can imagine, is quite expensive. So shifting gears a little bit, let's talk about microfluidics and how it can help us find the optimal sperm. So here, first of all, our goal is not to replicate the work of this company, which was interesting to say the least. You know, the semen analysis is not a perfect test. These are 4 editorials that I've written, basically talking about what the problems are with the semen analysis. And then, you know, there's also a lot of data indicating that total motile sperm count is a much better indicator than a lot of these other variables in a semen analysis. We actually do have WHO criteria for what is normal versus abnormal, such as concentration, count, morphology, motility. So one of the fundamental problems we have with the semen analysis is that it looks at a population of sperm, when what we're really interested in often in the fertility world is one sperm, especially when it comes to IVF or ICSI. So if you look at all of the available procedures other than ICSI, you use them to evaluate a population of sperm. So for ICSI, for example, we're interested in one sperm. And the problem is we don't have the way to interrogate a single sperm without killing it. And microfluidics really holds the potential to selectively sort, non-invasively image, and select one sperm. So when we look at sperm identification from testicular tissue, it's a really labor-intensive process for embryologists. Microtessie sperm identification can take 12 to 15 person hours, and the success of microtessie is really dependent on the lab. What if we could automate this process? That's kind of how I initially got interested in this. So the goal is to do sperm sorting on a chip. So essentially you put either the semen or the testicular tissue sample into a chip and then run it through this microfluidics chip, and then it sorts for the various, removes the debris and finds kind of the optimal sperm. So a little bit of background on microfluidics. What is microfluidics? It's manipulation of cells through channels with dimensions on the order of microns. So really, really tiny, tiny channels. It allows for really precise control of a cell's environment and manipulation. And then also it's really excellent for processing samples with low cell concentration and really low sample volumes. Another important point of microfluidics is that these devices can be run iteratively. In other words, they can be run through the device, and then the part that you want can be put back through the device to get even more purified. And we'll go over that in a second here. So how does this really work? There's a number of ways that can be done. A lot of the commercially available chips, such as the Zymot chip, rely on sperm motility to select the optimal sperm. We're really leveraging these microfluidic devices and with the idea of not relying on sperm motility because we're focusing on testicular sperm. So you have lift flow and Dean flow as these sperm flow through a channel. And the lift force really pushes the particles out. The Dean force pushes the particles in. And you get this dynamic balance between these two, and the cells sort by size and shape. This is an example that we've done. This was a review article. We published a couple of papers on this, looking at taking samples with a lot of white blood cells and sperm, runs through this spiral channel, and then due to the lift force and Dean force, they sort out in different directions, and there's two different outlets, the sperm going one way and the white blood cells going another way. And you can see kind of at the end of this channel here, you could run the good effluent, the sperm, back through the channel and get even more purification. So one thing to remember is that spermatogenesis is really high throughput. When we look at this, you have these spermatogonial stem cells making, in aggregate, most guys are making 1,000 sperm a second. And then you have these spermatogonial, you might have one spermatogonial stem cell that's kind of making the optimal sperm. And the question is, maybe it moves faster. Obviously a joke, I have ludicrous speed on here, but we don't really have a great way of identifying this sperm that might make the best baby. Certainly, it's hard to identify that in the lab. So when we have, the solution that we came up with is these spiral channel devices, and they're, etched in PDMS, and where there's a number of ways to make these. They're very inexpensive, like less than $1 to make. These are sort of the features of the channels that we're actually using. They're quite small, and all of these variables can be manipulated to optimize sperm sorting. And the key here is kind of getting these variables correct so you can sort the sperm in an optimal fashion and get the desired outcome. So when we look at applications for microtessie samples, the goal was to develop something where we could put the minced biopsy in and then actually have put the media in, and then have the sperm go one direction, the debris go another direction, and then run this through an enrichment module to further isolate the sperm. And these devices work with a syringe pump actually actively pushing and sometimes actively pulling to the sample through the device. So in doing this, the sperm search was accelerated about 10-fold, and this really could improve the sperm search success rate. The sperm was, this looked at a project to find sperm through microfluidics in samples from men with non-obstructive azoospermia or spermatogenic failure. We took these samples and ran them through the device. These were microtessie samples. And this was just from a research standpoint. We weren't using this clinically. These samples were a lot cleaner. They had a higher concentration of sperm found per unit time, 2 to 20-fold higher, on average about 8-fold higher. And it took about 90% less time to process. This is kind of the raw data here with our samples. And this is the paper that we recently published in Urology. And when we look at sort of our device versus a standard, centrifuge, you can see that our device puts 6.2 Gs of force on the sperm, whereas the centrifuge puts 500 Gs. We also are exposing it to that force for a lot less time. And then you can see, if you look at the bottom right, the ratio between the test and the control results, you see that it's about 8, almost 9-fold higher, meaning we're finding a lot more sperm. And in a few instances, we actually found sperm with the device where we didn't find it looking manually, which obviously could be a game changer for patients. So now we'll switch to sort of the final component of the talk and talk about spermatogonial stem cell culture and how that could really play a role in shaping the field in the future. I suspect we'll finish the talk a little bit early and then have a decent amount of time for questions after we get through this, because this is the final component here. So, really, what's the, one thing we can look at is obesity. I mentioned obesity at the beginning, and it can obviously have an impact on offspring health. And, so we started looking at this to try to understand, how we might be able to culture spermatogonial stem cells. One thing you have to understand is that while for a long time in rodents, we've been able to culture spermatogonial stem cells in the lab and actually grow sperm in a dish, essentially, even from iPS cells, we cannot really do this in humans. And that's probably due to the fact that spermatogenesis in humans is fundamentally different than in mice. So we started looking at this in about 2013 or 2014. And when we first did it, the work required 3 years and 8 fresh whole human testicles. These were from patients who were having orchiectomies for kind of the end of the road for testicular pain. Obviously, this is a joke, but you could volunteer for this if you wanted to. We now have a pipeline of cadaveric organ donor tissue, and we're getting about 40 to 50 specimens annually from neonates all the way to age 75. And that's really been a game changer, because we actually get, the whole pair of testicles from all these patients. We get them fresh. We obviously have approval to do this with the donor program. And when we look at this, what we find is that, we've sort of classified these cells into 4 different states. And the SSCA4 positive cells are kind of the spermatogonial stem cells that replenish other spermatogonial stem cells, while the KIT positive cells are the ones that actually make sperm. And we found that there's some unique chromatin features that drive this transition. And it's important, you might ask, why is this important? Well, it's important to understand this process, because one of the big issues when you try to culture spermatogonial stem cells is they all kind of become fibroblasts, and the tubules become really atrophic, and they don't really last very long. And that's the big problem. So you've got to look at what are the factors that drive pluripotency and sort of repress totipotency to understand how this works. And this is the basic protocol that we went through. We sorted the cells and then sequenced the cells. And then you can see here that if you look at how these cells journey through the process from the stem cells to actually becoming sperm, the self-renewing spermatogonial stem cells are SSCA4 positive, and then the differentiating spermatogonial stem cells are KIT positive. So those are kind of the differences that you see there. So what we found, which was really interesting, was that metabolic regulators really govern the spermatogonial stem cell differentiation and the renewal. And what's super interesting is that if you look at this cluster C, as these cells become, to the point where they're producing sperm and not just renewing themselves, what gets ramped up is a lot of the metabolic factors. And you can see that here. And then the cell cycle replication repair. And that really makes a lot of sense, because these spermatogonial stem cells go from just, reproducing themselves at a very low level to really ramping up and making a huge number of sperm. So if these metabolic transitions govern the spermatogonial stem cells, you could ask the question does the altered metabolome get recorded in the stem cells? And if it did, would there be evolutionary advantages to this? And the answer is absolutely yes. If you're going through a time of famine, obviously it makes sense to have children that probably have a lower metabolic rate. Could these SSCs serve as a tape recorder of nutritional exposure that's passed on to offspring? And if they do, what's the mechanism? Is it microRNAs? Is it methylation? Is it protamination? So next we published another paper in Cell Research in 2018. And this work was all done by Jingtao Guo, who is, was a graduate student with Dr. Cairns, who's the senior author on this. And Jingtao has now joined the faculty at University of Utah, so we'll get to continue this work. When you look at this, what you see here is we can look at the expression of all these different genes, and you can look at something called pseudotime, and that kind of clusters the cells based on what they look like. What we're doing here is single cell RNA sequencing, so we're profiling, 10,000 cells. Here was, 8,400, and we're looking at all of the gene expression from an RNA standpoint in each of these cells, and then we can see kind of how the cells look different. And what we can see here is you're essentially watching spermatogenesis. So the state 1 here are the SSC, spermatogonial stem cells, and state 8 are the sperm. So you can actually watch the transition here. And then when you look at these other cells, the 9, 10, 11, 12, 13 are some macrophages, endothelial cells, myeloid cells, Sertoli cells, and Leydig cells. And it turns out that the Leydig cells and the Sertoli cells and even the myeloid cells are really important to maintaining the spermatogonial stem cells. And we can derive a lot of insight from this in terms of how to culture them. So, when you look at this again, this is just another representation of kind of the different expression here and how we derive this. And this is, biologic replicates, and these are all done with technical replicates as well to sort of look at the different cell types and see how things are changing over time. So this is the single cell sequencing, and essentially what you're watching is kind of the birth of sperm through this, from SSCs to these differentiating ones. And you can see that these cells all cluster in different places based on their gene expression. So these are foundational data sets enabled by a cadaveric tissue pipeline, like we talked about. You look here and you can see further changes in the gene expression dynamics during spermatogenesis, and you can see how these things all cluster together and what the primary expression is in these different cell types. And what's really interesting is there's some key differences with the mouse, which will probably be the crux of understanding how to culture these cells. So when you recluster all these cells, the type 1 and 2, which are the SSCs and the differentiating spermatogonia, and to tie this back to the first paper, that sort of, state 1 and state 4, what we actually found is there's another type of cells that cluster called state 0, which are kind of the quiescent germ cells. In other words, they would be the germ cells that are there kind of, before you go through puberty and spermatogenesis gets turned on. When we look at these, what's really interesting is there's sort of a dynamic interplay with these cells kind of going back and forth and going from state 0 to state 1. And they can actually flip back and forth. So what we're trying to do for spermatogonial stem cells is figure out how to maintain the state 0 and state 1 cells in culture and then drive them to the state 4 where they can actually make sperm. And doing so requires a unique kind of combination of different growth factors and signaling factors. So the goal is to kind of leverage this bidirectionality between different cell types. So the next thing we did is we looked at human testis development during puberty. And this is again from this cadaveric organ donor pipeline. And we have, juveniles age 7, 11, 13, and 14, and then 25-year-old, and then also a 1-year-old. And we looked at their tissue, and now we're essentially plotting the same type of plots, but we're looking at how these differ between ages. And what you can see here is that, when you look at cluster 1 here, which are the, spermatogonia and the spermatocytes, you're not really seeing many of those until you get to until you get to, some of the older patients, and you do see a lot of these quiescent cells in the younger patients. And the goal is to kind of understand how to do this to eventually take, like, for example, prepubertal patients who are having testicular tissue bank for fertility reasons, freeze the tissue, and then in the future culture it and grow sperm in a dish. The other thing that you see here is that there's a big change in how the Leydig cells change between the younger patients and the older patients. Same thing for Sertoli cells and same thing for the myeloid cells. And one of the key insights we've taken away from this is that it's actually these cells that sort of are critical to maintaining the germ cell niche and facilitating spermatogenesis. Some other interesting things we've seen is we do a fair amount of transgender surgery at the University of Utah that's done with myself and Jeremy Myers. And then we actually see that, you know, you can, if you look at patients who've had their testosterone suppressed for, you know, 5, 10, 20 years in the transgender population, the tissue looks a lot like, you know, prepubertal testis tissue. And you can see that, you know, you can rescue, if you're trying to culture these Sertoli cells, you can rescue them with testosterone supplementation. So if you have the base medium here, you look at culture at 1 week, 2 weeks, and 3 weeks, and you see a lot of sclerosis in the cells and things sort of dying and not maintaining the architecture. Whereas if you culture them again and add testosterone, it actually looks much more like a healthy tubule, which is what you start at. So this resulted in, you know, the one paper we talked about here. This was the adult human testis transcriptional atlas, and then this is the sort of dynamic transcriptional cell atlas of testis development through puberty that we just discussed. So in some sort of 3 papers, and then there's another paper under review, and hopefully we'll be able to get some more grant funding to continue this work. So what does this mean for the field? It means a couple things. First of all, it really holds the promise of identifying and correcting spermatogenic defects and the impact of exposures in vitro. If you can culture spermatogonial stem cells, you can, you know, understand what's wrong with them. And for example, an azoospermic man, and you can actually potentially manipulate that through something such as CRISPR. And it's much more comfortable for sort of most people to manipulate spermatogonial stem cells rather than an embryo, which obviously has been incredibly controversial. And then, you know, the race is kind of on to see who will get there first. This is a long-term project of our group at the University of Utah and other groups as well. Just to bring it back to practical relevance, this is a Japanese group that's done ROSI, or round spermatid injection. So essentially in patients who have a microtesty, you know, decent amount of the time you don't find sperm, but sometimes you do find spermatids, and they actually took these spermatids, which are basically immature sperm that have gone through, you know, meiosis, so they're haploid, but they haven't fully developed into sperm, and they injected them. And amazingly, it actually works, not a lot of the time, but sometimes. They have 90 kids born due to this, and they're pretty healthy overall. They found these spermatids in about 10% of men after a failed microtesty, and then when you use them, they had about a 10%, you know, live birth rate from these. So very, very low birth rate on the order of, you know, 1%, but the kids appear to be pretty normal. And one thought, you know, would be it's really hard to recognize these spermatids, and could these be early spermatids which have not gone through meiosis and not late spermatids, which is what you want. And obviously the single cell RNA sequencing can be a great tool to understand this and to kind of figure out how to, you know, improve this process. We could also look at extended embryo culture. There's a trial of this at EVRMA at RMA New Jersey, which is a group I also work with. So I will at this point, you know, sort of take any questions, and then we can wrap up. And if we don't have any questions, we can wrap up a little bit early.
Okay, thank you, Dr. Hotaling. Interesting talk. I have one question for you, actually. So circling back to microfluidics, are there any potential sperm characteristics that we can use microfluidics to select for those sperms? And then is there, or are there any effects, known or potentially unknown effects on the sperm when the sperm goes through that microchannels in the microfluidics process?
Yeah, that's a really great question. We looked at that with ours, and we looked at, you know, viability, you know, motility, and, you know, DNA fragmentation, and we didn't find significant changes. Other groups, you know, such as the Zymot chip uses basically a microfluidic swim up, and it has shown that they can successfully select sperm that have lower rates of DNA fragmentation. you know, the jury's kind of still out until you have large numbers of kids born. After using these chips, you won't totally know the answer to that. It seems to be generally safe, and I would sort of submit that it's likely safer than a lot of the density gradients that we do.
Interesting, thank you. So you can use the Q&A box to submit your questions. And then if we don't get to the answers, you can find the answers on the website. Meanwhile, please fill out the lecture evaluations. You can scan the QR code here or just go to the website. Recorded version of this talk and the slides would be available online on the website. So I don't see any other questions, and I think we can wrap this up.
All right. Well, thank you very much for giving me the opportunity to talk today. And yeah, if you have other questions, let us know. Thanks.